Welcome to Biology of Marine Mammals. I'm Dr. Leslie Williams, and this entertaining lecture covers marine mammal reproduction. In this lecture, what we're going to do is we're going to look at some of the trends of mammalian reproduction and how that poses some challenges for marine mammals as they try to reproduce in an aqueous environment. We're going to look at two of those challenges: anatomical drag associated with external genitalia, and then trying to internally fertilize during copulation in the water. So to start, we first revisit the idea that there are several reproductive trends in mammals. So for example, as a generic rule, when you're looking at mammals, the male genitalia, particularly the scrotum, is external. Fertilization is always internalized in mammals. Gestation is also internalized in the female. Females give live birth, and then finally females lactate or produce milk to nourish their offspring. In this first part, we're just going to look at challenges 1 and 2, so the male genitalia and the fertilization, and some of the adaptations that marine mammals have adopted to deal with those challenges. So let's go ahead and start with the external genitalia. So in a previous lecture, we learned that mammalian males keep their testes external to the body in a scrotum. And this is because the testes actually need to be a little bit cooler than the body temperature to promote sperm development. The problem is that when you have these external appendages or protrusions, then you're going to introduce a lot of drag in the water. So in other words, if a dolphin, for example, were to have an external penis and testes, then while the male dolphin was swimming, that set of appendages would create a lot of anatomical drag, and therefore it would make swimming a lot more difficult or efficient. So therefore, to prevent that drag, many marine mammals have come up with a solution, which is simply to internalize all genitalia. Now, when we take a look at cetaceans, for example, we see that the male genitalia has been internalized, as have the female genitalia. So when we look at the females, in fact, if you've ever wondered, how can I tell a male and female dolphin apart when I can't see any of the external genitalia? The answer is actually in the positioning and number of slits. Females use what's called a genito-anal slit, and we can see that here on the screen. This is actually a harbor porpoise, and what we're looking for is a very long slit that contains both the vagina and the anus openings, separated from the umbilical cord by a great distance, which is over here. So there's where basically the belly button would be. And then in males, they actually keep their testes internalized, and their penis is going to be retracted in the genital slit. So the testes will be internalized and will never come out, but the penis is going to stay retracted right in here. Again, a short distance from the umbilical cord here, and then the anus is actually in a completely different location. So they have a distinct slit for the genital, and then again for the anus. As we see in this video of a orca, we can take a look at the slit and try to determine if it's male or female. And again, what we're looking for is how many slits do we see, and what is the position relative to the belly button. And we can see in this video that it's really going to be a female because she's got a very long slit, which suggests genito-anal slit, and then again a very far positioning back behind the belly button. So that explains what happens to the penis and the testes, but one of the problems that we already mentioned was that testes need to be external because the sperm actually require a lower temperature in order to mature. And so most mammals, if they have internalized testes, their testes actually get too warm, and therefore the sperm don't develop properly, and therefore they're not going to be reproductively successful. So how do the cetaceans solve this problem? And the answer is actually in using a countercurrent heat exchange system, something that we introduced in a previous lecture. So what they do here is, yes, they do have internalized testes, as we can see here, but they're going to wrap it up with cold blood so that it actually cools the testes down by a few degrees to allow those sperm to mature properly. So where do they get that cold blood? They actually get it from sending warm blood up to a different appendage, such as a dorsal fin or even a fluke. And then as soon as it cools off, and you can see here the radiating heat, then it basically comes back down and immediately wraps around the testes to provide essentially a little bit of a mini refrigerator, what I call the testicular cool down. Cools it down by just a few degrees so that those sperm can mature appropriately while still keeping the testes internalized. So now that we've finished with cetaceans, let's take a look at the pinnipeds. Now the pinnipeds actually have some differences based on whether or not they're phocids or otariids. Phocids, again, are going to be the true seals. And in these guys, the testes are truly internalized. In otariids, however, they actually do have a small external scrotum. It's not terribly large, but they do keep their testes more externalized. So again, the phocids, the internalized testes, probably use the same countercurrent heat exchange system, and the otariids look more like traditional mammals with an external scrotum. In both genera, however, we're looking at basically the fact that the penis is going to stay internalized, in part, again, to prevent that anatomical drag. Now, what's interesting about all pinnipeds is that they use a special penile bone. It's called a baculum. The baculum actually used to be collected by seal harvesters, and they would actually use it for decorations and things. So it's not uncommon to go to maybe a native museum and actually see carved baculum, which is essentially a penile bone. Of course, humans have no such thing, but a lot of animals do have this, and that includes the pinnipeds. And so you can see up here, the top is going to be a walrus baculum, and this is one from a gray seal down here. So this is a bone that allows them to maintain an erection when they actually are ready to copulate. So now we're going to turn to our next problem, which is maintaining internal fertilization. Now, when we look at most aquatic animals, and this includes the aquatic invertebrates, and this includes fish, most of them use what's called external fertilization, whereas a female will lay a bunch of eggs and then pretty much leave the eggs for a little bit as the male comes and just sprays sperm all over. That process is sometimes called spawning. Now, on land, internal fertilization is pretty easy, in part because the female, when she is mounted by a male, uses the ground as a stabilizer. So now when we re-enter the aquatic environment and we're looking at the fact that mammals are forced to still use internal fertilization and there's no ground for stabilization, you could understand why maybe it's a little bit of a challenge for example, a male dolphin to successfully inseminate a female dolphin in the water where there's nothing to stabilize against. So how do they do it? Well, in reality, it's not easy. So when you look at cetaceans, for example, who have no choice, they must copulate in the water because they never come out of the water, they have several adaptations to try to promote successful insemination. One of which is that copulation is very quick and then also repeated multiple times. So how quick? Like 5 to 20 seconds quick. And of course, this is going to be after a huge mating display or courtship display. So they might be courting each other. A male might be courting a female for a very long time, but the actual copulation is very quick. A female will turn on her side a little bit. A male will try to kind of come up next to her and quickly insert his penis into her genital anal slit and hope that it's successful because, again, they can't really hold that position for very long. Also, very interesting, the testes are extremely large. Now, interestingly enough, the larger the testes, the more sperm is created because that's where sperm is created. And therefore, the more sperm will be delivered during copulation attempts. So we're actually going to look at testicular size here for a moment as anatomical adaptations to different reproductive challenges. And I'm going to go ahead and start with not marine mammals, but rats, because they display the most interesting mating strategy. Now, in this picture here, you can see that we have a male and a female rat. I think it's pretty obvious which one is the male. They have extremely large testes. Now, rats, of course, are not going to face the same problems that marine mammals face, but they have a really interesting mating strategy, whereas multiple males will inseminate the same female. And so the most successful male will be the one that delivers the most sperm. So therefore, there is a sexual selection towards larger testes because he who delivers the most sperm gets to create the most offspring. So essentially, he who has the largest testes gets to be the most reproductively successful. So there you can see basically a selective force towards larger testes. Now this isn't really the case in cetaceans because we're not looking at multiple matings like this, but really the large testicular size reflects the fact that it's very difficult to successfully inseminate in the water, and you just need a ton of sperm in order to do it. So how big are these testes? Huge. For example, harbor porpoises hold the mammalian record for the largest testes as a percentage of their body weight, a whopping 5%. And although it may be a little bit difficult to see in this picture, this is basically a bisected harbor porpoise. This right here is going to represent the one of the two testes. 5% is a huge amount. I don't know what it is for human males, but I would estimate that it would be more like 0.1% or 0.2% of their body weight. It's not huge. And so the second place winner is actually the right whales, which each testes can weigh over 1,000 pounds. So think about that for a moment. That's an extreme amount of testes. Again, reflecting the fact that fertilization in the water is difficult. All right, so now we're going to go ahead and turn to the pinnipeds. Now the pinnipeds, even though they usually have the option of land, some do copulate in the water. But what's interesting about the pinniped copulations is that they actually have that additional penile bone, the baculum. Now the baculum is thought to help them achieve successful copulation. It allows them to maintain an erection and also keeps the penis in place, again, to ensure proper sperm delivery. And as I mentioned earlier, baculums are not that uncommon among males, but pinniped baculums are larger than most. Which raises an interesting question. If baculums are so useful, how come cetaceans don't use them? And that's a really interesting question because all fully aquatic marine mammals lack a baculum, so this includes the sirenians, but it's possible that a baculum would actually be dangerous when trying to do a water copulation. In other words, if you take two dolphins and one dolphin is using a very stiff bone to keep his penis erect, you could imagine that if he misses or somehow gets a little bit out of place during the insemination attempt, that baculum could actually tear the vagina in the female, which would at worst make it not reproductive to the female, but it could actually kill the female as well. So that's one possible reason why baculums are not found in male cetaceans or sirenians. Anyway, that concludes our lecture on some of the anatomical challenges with reproducing in the water. And with that, I'll see you next time.